Hello. Mansi, it took me a while to get on. Was there some problems before, or have I come in late?
No, I couldn't get on either.
Okay.
Can't hear you, Mansi. I don't know if you can hear us. Too bad.
Okay, I'm glad that not everyone's Monday has started like mine. Apologies again, and let's start, and let's not waste anybody's time. Today we are going to talk about amazing mitochondria, and it's a short lecture. I have kind of tried to keep basic so that when you go in the clinic, you realize that at this level, sometime, when you're not finding what's happening, try to explore this area, because I'm sure in coming years, this is going to be an area of amazing, what do I say, clinical judgments. And the reason I've got this pre-lecture activity slide open is whenever you have time, have a look at this first video for sure. It's only a 40-45 seconds video. And this will show you how the mitochondria are involved in an amazing set of activities about which we are going to learn today. So mitochondria, for a long time, they were primarily considered to be the powerhouse of the cell, producing the energy required for cell metabolism by oxidative phosphorylation. But now what we are learning is that they are involved in numerous other physiological processes, and they are actually very dynamic. What do we mean by dynamic? As the slide is commenting, that they are doing something we know as fission and fusion. Okay, it has nothing to do with nuclear fission and fusion, but they have their own way of what I see as dancing. They are changing shape when they are dancing. They are exchanging material, metabolites. They are changing size also when they are doing that. And while doing so, while dancing, they complete all the amazing work they do. What this slide is also showing you is the structure of mitochondria, which I know you would remember even from your primary school days. We used to make this oval shape and then draw these crystals inside. But now we know that actually they have got two membranes. There's an outer membrane, there's an inner membrane. Activities are going in both of them. Plus, there is a lot of activity going in between these two membranes also. And can you see something else? DNA. What does that mean? Would anybody like to comment?
Is it that mitochondria has its own particular DNA, and that's how you get that maternal mitochondrial DNA, and the theory of you can trace it back to the first female in history?
Wow, Mary, was that Mary?
Yes.
Wonderful. I'm sure you have read some of this amazing stuff. Absolutely right. So mitochondria have this spatial DNA. For a long time, it was thought that it is derived only from maternal side. However, that theory is kind of no longer valid. We are now noticing that a portion of it is inherited from our paternal side as well. Okay. So you can imagine that this DNA is like any other DNA. It will be susceptible to pollutants. It will be dividing. Okay? And the way mitochondria dance around, the way they complete their fission, fusion, change of shape, size, that decides how well body's processes are happening or functioning. So what do I mean by that? What body processes would mitochondria taking control of? Anybody? Okay, we have somebody in the chat box. Yes, Tracy, ATP production is the main activity they are doing, but in doing so, they are actually supporting various aspects of our human body. So let's kind of elaborate on this dancing dynamic activities. One is fusion. That means when two organelles, when I mean organelles means two mitochondria, they join. Fission is when they separate. Transport is when they are themselves moving around in the cytoplasm, or they are helping somebody else to move. And I'll expand that later in the slide. Now look at the fourth dot point here. What does that mean? Again, I would like somebody else to share if they have read anything like that. Again, I pronounce it in two different ways. I can call it mitophagy or mitophagy. What do we mean by that? So there is a hint in the brackets. Yes, James, breakdown of cells, and let's be very straightforward. It's actually killing of the cell. Or auto means cell, phagy means eating. So literally it means self-eating. And when mitochondria are eating themselves, instead of autophagy, we call them mitophagy. Okay? Do you think this could be of relevance? Maybe just tying it up with whatever we know so far of fasting. And I know we discussed a little about it in one of the sessions earlier also. So Mary is with me. She agrees that it might be of some importance. And Tracy is saying perhaps, maybe when the mitochondria are damaged. And Dale is saying it is important, but that would decrease ATP production. In my understanding, in a normal person, this is a normal process. When something is damaged, body or mitochondria are capable of eating away that molecule, that mitochondria, and in fact, they utilize the protein or the fat or the carbohydrate component of that molecule for some other anabolic pathways in the body. Again, I would not go into detail, but I just want to open that window for you people that when that autophagy happens, it initiates a signaling response in our body that actually help us fight certain disease molecules. So in a way, it is a prevention of apoptosis, which when, not prevention, but it is doing apoptosis in a controlled manner so that we don't have a tumorigenic behavior. Okay. Dale is trying to clarify that it is not the mitochondria breaking down, instead mitochondria is breaking down something else. Let me clarify, it means one mitochondria is kind of self-dissolving itself, and in doing so, it is giving a signal through producing some molecules that can now be used by some other mitochondria to maintain their health. And plus, what Tracy was also saying earlier, mostly the damaged one, they kind of self-destruct, okay, so that they don't become harmful for themselves or the cell or the body. Okay. Now, as usual, I have added a complicated table here for you, which you might not be able to read properly. My only aim is for you to just look at the heading. Clinical syndromes due to mutations in gene encoding, fission and fusion machinery components. So that's where our understanding is moving now, that most of the underlying reasons for any disease condition could be simply a mutation in the processes. That is, when mitochondria are inhibited from doing their dancing. That's where our new understanding is moving ahead. Now, Dale has another point that if there are less mitochondria, would this lead to a decrease in ATP production as they are the ATP producers? Dale, if only a damaged mitochondria has dissolved, and there are a number of mitochondria, okay, this process should not affect ATP's production under normal circumstances. However, as we will explore later, if somebody is kind of living in an environment where they are exposed to a lot of environmental toxins, xenobiotics, that is maybe harming the mitochondrial DNA, and then initiating this death of mitochondria at a higher speed. So that's not normal. And yes, then you will notice that people are complaining of fatigue because their mitochondria are not sufficiently healthy and they are not sufficient in numbers to produce ATP. Okay. So couple of slides to understand the structure of mitochondria a little more. So number of mitochondria per cell, kind of connecting to what Dale was just saying in the chat box, that number of mitochondria per cell is dependent on the function and energy requirement of the cell. So through the process of evolution, through our childhood years, the cells that require and expand most energy, they contain the highest number of mitochondria. Now we were talking about that it has two membranes. There is one outer membrane that contains porin, kind of a structural component that ensures that there is a porous environment in that outer membrane. Why porous? Because all those metabolites that we generate from carbohydrate and fat and protein, they have to enter so that our Krebs cycles or oxidative phosphorylation or ETC can take place inside the inner membrane. Inner membrane, it contains a phospholipid, which is called as cardiolipin. It is highlighted in red. So I'm sure now you would know that this is the name I would like you all to remember from the exam perspective. Okay. So very important that we have healthy mitochondria and healthy membranes of mitochondria. And how do we ensure that? By ensuring that there are sufficient phospholipids, because they are the components of that membrane. Mainly, so far we know that it is made up of phosphatidylcholine and phosphoethanol amine. So what does that tell you? If there is sufficient amount of phospholipid in that membrane, then only that membrane will have integrity. It will be agile. It will be appropriately permeable to allow communications that are happening between different cells. And components of ETC, they are all located on the inner membrane. Then between these two membranes, we have cytochrome C. Does that name sound familiar? Cytochromes. Okay, thank you, Jane. So, yes, Dale, thank you. So, they are cousins of the cytochromes in the liver. There are many cytochromes in our body. So, yes, Dale, this is the mitochondrial version of cytochrome, and we call it as cytochrome C. Let me just focus on a couple of chat messages. Yeah, so Dale is sharing a reading from a journal article where they talked about how faulty mitochondria are correlated with aging and diseases. Thank you. That's really helpful. And I would encourage everybody to go and review that article. We will also discuss that information at the later part of our lecture today. Tracy has a question. So, all are the mitochondria numbers per cell individualized? Do more active people upregulate their mitochondrial content? Very logical question, Tracy. I don't remember reading that thing directly anywhere. So I'm not sure But looking at cellular level, because they themselves, the mitochondria themselves love that activity, and if they are happy, I'm assuming their number will increase if they are allowed to be happy and dance and do fusion and fission, their numbers would be healthy. But I am not sure whether this has been measured as yet in people per se. It has been measured in some of the tissues and organs, as I'll comment later in the slide. D is saying that more damage, poor functioning mitochondria from illness or poor diet will be equivalent to poor body functioning owing to lower ATP production. Absolutely, D. Absolutely. And this is actually asked in the exam sometime. What do you mean by mitochondrial dysfunction? And I've noticed that sometimes students, because of the exam stress, they don't give that direct answer. So thanks, D. Yes, that is what it means. Dale is asking one more question. Do we know if the mitochondria has a flow in certain direction in the matrix? Have a look at that short video, Dale. And because I again don't have the answer to this, sorry. Okay. Really don't understand that one and can't even speculate from whatever I've learned so far, whether, you know, it is actually possible that intuitively, you know, they would migrate towards a particular side, and it will become clear when we look at their functions, okay? So I think you may be right there, but let's wait till the slides on functions appear, okay? Right, Stephanie also has something amazing to share with us. She's reading a book that suggests mitochondria amount in the muscle cell increases when there is higher demand of energy. Okay, cool, good. Share that book link and also try to find out from where they are quoting that information. You know, sometimes they are quoting that information from a study done in a rabbit or a sheep or a rat study. So just pushing you towards have your critical cap on your head as a nutritional biochemist, right? Okay, let's I'm so happy that we have so much discussion going on here. Okay, good. We have many like-minded people here. Okay, good. Good. You people seem to have read a lot more than me. I have not read that those books or anything. Okay. Maybe that this kind of will give you an indication that the number of mitochondria can vary from 200 to 2000 per cell. Muscle, liver, brain have higher level. Oocytes have up to 100,000. Can I ask you people to speculate how many sperm? How many mitochondria could be there in a sperm? Just a speculation. And
I'd say perhaps more, wouldn't it? Because they have to be motile. They're motile, whereas like an egg is immotile. It's just, you know, flouncing around in there. So I'd say it would need more mitochondria to power, maybe. That's what I think.
Yeah. I also thought so, but I am relying on the last link on this slide for this information. And it told me, I've written the number in the chat box.
Well, that's a surprise.
Yes, it is. It is. Now, what do you take away from your clinic perspective from this particular information? Yes,
Jane's point seems on the mark, doesn't it?
yes, yes. So again, think in perspective of your clients. I'm not sure if you have noticed that fertility issues have also kind of increased in our community. More and more people are finding it hard to conceive, and one of the reasons, because it is such a multifactorial issue, one of the reasons that we are now finding out is actually the health of those sperms, which you can see because they have so low number of mitochondria, which you can think the potential of those mitochondria, because they are such a fast mover, as Mary suggested. How important is that number of mitochondria relevant? And then look at that. They have only one copy of mitochondrial DNA. That means if there has been any exposure, to toxins, to pollutants, to stress even, this DNA will get damaged very quickly. So again, we are again still understanding these processes. That is one interpretation we can make and be prepared to focus on such dietary interventions for client as well. Even talking about the environment and how to keep it mitochondrial friendly. Okay. Couple of slides to talk about mitochondrial functions. So the first one, numerous biochemical processes are taking place here. Oxidative phosphorylation, Krebs cycle, beta oxidation of fatty acid, calcium handling, heme biosynthesis. So various crucial mechanisms are happening here. Therefore, dysfunction of mitochondria is associated with various disorders, metabolic diseases, cancer, neurodegenerative diseases, aging processes. I'll just comment a little bit more about its role in central nervous system. So there is evidence that alterations in the mitochondrial morphology. So when I say morphology, what I mean is their fusion and fission mechanism that influences brain energy metabolism. And mitochondrial activity is now observed to be involved in the pathophysiology of neuropsychiatric conditions, because they are giving energy to the brain cell, to the neuron. So if they are not functioning properly, obviously the cells are going to die off. Cells are going to be fatigued, and they are not going to be involved in the signaling pathways that we learned about in the last session. Patients with depression, they can't utilize glucose sufficiently in the mitochondria. Okay. Now, this is another important slide with regard to functions of mitochondria. So again, just kind of bringing your attention that in your final exam, you may be asked to give a short answer to the functions of mitochondria or how they prevent disease processes. So these two slides are very important from that perspective also. Read it and you explain. One of you explain what is this slide conveying to us. Just last line, sorry, last dot point is the take home message. And something what Dale was commenting earlier, maybe you can correlate to that, whether their activity is diverted or focused to a particular section of the matrix. Is it too difficult? Yes, Jane, absolutely. You are spot on again. Yes, that's kind of the summary. We will, that's the reason I have a couple of slides to refresh your memory about the Krebs cycle. I know you people have studied it in the bioscience. I'll expand on that when we reach there. But what this slide is saying is that whenever there is an attack on our immune system, which can be viral or bacterial or whatever, you know all that, then those antigen presenting cells that sense that this molecule is an antigen and it's there to harm me, they actually start sending that material to the mitochondrial membrane. They divert it there. And then mitochondria uses its intermediate compounds that it is producing when citrate is going around in circles. Those intermediates are used to fuel up our immune cells. So in a way, our immune system or our immune cells are telling mitochondria to pump its section, bring it on. We need those metabolites. We need to fight out these factors. And how do we know that? Because there has been some animal experiments where they withdrew citrate and succinate and fumarate from a particular cell, and then they saw that the activity of the immune fighters went down. So why mitochondria are doing that fusion and fission, now you would understand. It's their way of taking message from one area to the other. So they are taking the message from immune cell, going inside, telling the Krebs cycle to pump it up, and then taking those metabolites back to the immune cell. This is a summary of how mitochondria are considered central to diverse kind of pathways. It's not only biosynthetic pathways, but bioenergetic pathways, maintaining a balance between free radicals and antioxidants. It is actually controlling apoptosis. That's kind of a summary of the amazing work that they do. Dale has another point, is that because the cell membranes of the dendritic cells and macrophages are not produced properly. Yeah, because the cell membranes are not healthy. Yes. If they are not healthy, this signaling is not happening for sure. Oxyphos means oxyphosphorylation, and MET means electron transport, mitochondrial electron transport. In fact, just one little more thing, which is extra, that when this apoptosis is happening, this is happening under the action of certain enzymes. They are called as capsaicins that are released by mitochondria. That deeper is the connection of mitochondria taking control of apoptosis. So they kind of give a signal if it is not working properly. Okay. So definitely, like myself, you all now must be wondering how to ensure that mitochondria continue to dance. We want them to do fusion and fission. We want them to be active. We want them to do all kinds of dance, classical, jazz, whatever you like. So several micronutrients have now been identified that can support the pathways that are happening inside the mitochondrial membrane so that they ensure that ATP is produced optimally. And most of the nutrients that are listed in the first dot points are actually those ones that support TCA cycle. Your B vitamins, carnitine, lipoic acid, alpha tocopherol, that is vitamin E, because it is so important to prevent oxidative damage to the phospholipid layer. Selenium, as you would have guessed, is because it is anti-inflammatory. It is supporting glutathione, which is the major antioxidant for the ETC pathways. Now there is something written in the second dot point. Melatonin. Where does melatonin comes here? Has anybody read about that also and would like to share with us?
Well, melatonin's your sleep hormone, isn't it? So is it saying that whilst you sleep, it powers up the mitochondria?
Could be.
I was just throwing it out there. It doesn't have to be correct, but I just know that the melatonin is what happens. That's the hormone that, you know, makes you sleepy.
Yeah, I appreciate that. That's why I'm not categorically saying that you are wrong, because that's a very rational thought. But I don't have evidence to back it up. It's making a sense to me also. What I have read is that melatonin is also acting as antioxidant. Okay? And it has actually emerged as a potential therapeutic tool for treating Parkinson and Alzheimer, just because it has an antioxidant role to it. Whether it is via the pathway that you have commented, I'm not sure. It is a possibility. Okay? So I like the way you're making association. Yeah. So that's the reason I read about, which is a possibility. Few other nutrients. Again, I encourage you to remember some of them for a short answer question. Then as a case study also, you might notice that this case has a poor mitochondrial function, and then there would be a short question that will ask you which nutrients you will intervene with to support this particular plant. So from that perspective, and then in your prospective clinic, or if you choose to write a blog item, you would be exploring these amazing micronutrients and how they are supporting mitochondria. We have discussed about carnitine. We know selenium is kind of an antioxidant, but some recent data suggests that when mitochondria are doing their biogenesis, that is those fusion and fission, selenium is helping them. All other nutrients that are participating in TCA are imperative because they are ensuring mitochondria is healthy and it is producing ATPs. Omega-3 makes another sense because they are so vital for the membranes of mitochondria, which usually nobody talks about so far. I'm sure I have talked enough about glutathione. So that's another one. This slide is kind of trying to refresh your memory of energy pathways, that when we are consuming proteins, carbohydrate, and fats, they all these pathways, they kind of pull at or converge when they produce pyruvate. Then from pyruvate, we can make acetyl coenzyme A, about which we discussed last week also, because from here we can make neurotransmitter acetylcholine, or this acetyl coenzyme A can be converted into citrate. And actually we are making this acetyl coenzyme A so that this is the molecule which can go inside the mitochondria. Pyruvate is a big molecule. And here you get another hint. Have you heard when people are exercising after a long break of months, which happens with me when I'm stressed and I don't exercise, and suddenly I wake up, I need to walk or do something, and then I have this muscular cramping in my calf muscle. And I'm sure you would have read about it, that one of the reasons is accumulation of lactate. And the reason is that because of some reason, this pyruvate is not going inside and not starting this cycle. Just one of the reasons. It is so, there are so many other layers to it. So again, to emphasize that during these pathways, we need all of our vitamin Bs as a big happy family supporting each other. That is kind of a main area where any kind of insult, any kind of inhibition can lead to dysfunction in the pathway, may not go further, which will mean no production of ATPs. Okay. This is the point which I was saying in that summary diagram when we started talking about energy pathways, that glycolysis takes place in the cytosol, then TCA takes place in the mitochondrial matrix. Glucose starts with one molecule of, glycolysis starts with one molecule of glucose, and we make two pyruvate molecules plus some NADH molecules, and all that then go on to start TCA cycle. The enzyme that we need here is pyruvate dehydrogenase, which in turn needs a couple of nutrients. Okay, I'm not going there yet. Right. And in the process, we are generating few ATPs. We are also then generating NADH and FADH. And then we take these NADH and FADH to ATC. Okay. At this point, let's take a 10 minutes break. I know this cycle always gives a headache. So let's take a 10 minutes break, and when we come back, I'll talk a little about this slide, and we will then move on to ETC pathway. Okay, we are back after doing a little bit of stretching, finishing up work, having our breakfast. I said I'll talk about this slide a little more. Just to refresh your memory that via glycolysis also we are generating few ATPs. Then in the ETC, Krebs cycle, we are generating few more ATPs. And from exam perspective, you do not have to remember that number. But remember that in the process, we are also making NADH and FADH, and we are making that from lysine. So these are the active forms of lysine and riboflavin. The thing is, can you see that they are carrying some hydrogen molecules? Hence, these molecules have the potential to generate more ATPs, because finally, what is ATP? It is energy, it is those electrons or hydrogen molecules. So it's very important that along with the metabolites, these molecules travel to the next stage. They have to enter electron transport chain. And as the name suggests, it is simply transport of electrons. So in that membrane of mitochondria are 4 sets of enzymes. And I have actually included a couple of pictures of the same diagram, just to ensure that you refresh this properly and remember it from the exam perspective also. So we have these complexes which we call as cytochromes, complex 1, 2, 3, 4. And with the support of these enzymes, we are just dancing, again I'm using that word, we are just throwing, we are juggling electrons from one place to another. And in doing so, can you see NADH is getting converted to NAD, FADH2 is getting converted to FAD. So where is that hydrogen molecule going? Okay. It is helping to produce electron, that is the energy, ATPs, plus we are producing water. Okay. So this in a way is the second stage of mitochondrial activity. And remember that B2 and B3 are so important here. That's why micronutrient deficiency plays a major role if somebody is having fatigue, lower ATP production. Remember what is the output of this ETC. It's so simple. It's just water and ATP. That's it. Okay. Now have a look at the 4 complexes that form the enzymes in the ETC, which are present on that mitochondrial membrane. So first complex, sorry, I think earlier I called them cytochrome complex. Just remember, the first one is complex 1, which has NADH, coenzyme Q, oxidoreductase, and it also contains iron sulfur protein. Remember when we were doing your first assessment, some of you commented on role of iron in fatigue, and this was the word I was looking there. Iron sulfur protein are so important for the functioning of complex 1. Complex 2 is succinate dehydrogenase enzyme, which also has iron sulfur proteins, and cytochrome B is present there. This is the complex which has ubiquinol, and ubiquinol is kind of a bridge from complex 2 to complex 3. Don't stress out thinking that you have to remember the name of all the 4 enzymes in systems. Maybe please remember ubiquinol, where it is important and what is its role. So Mary is confirming something, B2 and B3. Yes, niacin and riboflavin. Correct. Did I say correct? Did I earlier say B1? Okay, maybe 1 coffee was not enough for me, sorry.
No. It was, I didn't see the vitamin and I didn't know if I was getting confused with something else. So I just wanted to confirm that in slide 23, it was vitamin B2 and B3. So yes, you confirmed it. Thank you.
No, you are correct. I should write it as vitamin B and B3 because in this subject we use so many abbreviations. We should be careful. I'll correct this one. Okay, just one more important thing to say here. Again, I've added another picture of the same chain just to show that the way we are bouncing or juggling hydrogen in between these complexes to generate ATP, it is quite complex. It's happening at a micromolecular level, and it is a very sensitive reaction. And the last complex, it also needs copper. Okay, just kind of I cannot even call it a fun fact. It's an extra fact. Remember, there was a time in history when people used to use cyanide for poisoning. I know, such a weird thing to say in a lecture. The reason that's happening is because cyanide, it interferes with copper here at this level. So it prevents the complex 4 to act properly. That's why it leads to immediate death. Okay. Sorry, you don't have to remember it. I read it a long time and I always, whenever I see copper here, I remember it. And I can't help sharing that info. Right. So I hope now so far, most of you now understand how critical micronutrients are and how important mitochondria is for functioning of most of the biochemical pathways in our body. Now, a couple of slides to review what does the recent evidence say about the form of diseases that can develop into mitochondrial dysfunction, which is simply not production of ATP or lack of nutrients or when the membrane is not healthy. Hence, recent decades have seen a rapid increase in reported toxic effects of drugs and pollutants on mitochondria. In fact, there is a documentation that genetic differences leading to mitochondrial diseases are also noticeable. One in about 4,000 people would have some gene environmental interaction that lead to mitochondrial damage. Again, I'm not sure whether it was originally there in the genes or whether the way we have changed the environment that has led to that state. Now, some of the other causes of mitochondrial damage could be toxic metals, some organic pollutants, plastics, some prescription drugs, alcohol. Normal aging could also be an issue because that may accumulate in mitochondrial DNA and prevent its further growth. I would also like you people to recall, remember when we were studying oxidation in session 9, we commented that we are producing maximum free radicals in this chain. So if we don't have a balance of endogenous and exogenous antioxidants, then we are potentiating free radical damage to the mitochondrial health. So that could be other factors. Some drugs, like I tried to look at which drugs, so mainly steroids, barbiturates, anesthetics, statins, fibrates, some beta blockers. These are some of the medications that have been observed to impact on mitochondrial health. So what happens then? There is decrease in mitochondrial function at the molecular level because those electrons are not allowed to dance around properly. They are not able to do their juggling in that membrane, which leads to low production of ATP. So that's kind of the only way so far we know that person is fatigued. This slide is listing some of the symptoms of mitochondrial dysfunction. And you may notice that they seem like too general because they can be associated with some other etiological factors. But having learned a little bit more about their structure and function today, I hope you can make that association. So if somebody is having learning disabilities, it could be iron deficiency, it could be genetics, it could be neural function, but at the core of it, it could be simply mitochondrial health. Have you, any of you, already heard Professor Terry Wall? I know I have given a link to her old TED talk in the post-lecture activity, but mostly students have listened to her so far. Yes, thanks, Jane. And it's such a eye-opening talk. So if you haven't, please make time to watch it.
Sorry, Nancy, can you repeat who that was by? Sorry.
So that's Professor Terry Wall. The link is given under post-lecture activities.
Okay, thank you so much.
And I know she was in Australia last year, I think, talking in one of the conferences to which I could not make it. Okay. Hence, so much interest and so much evidence now of association of mitochondrial dysfunction with Alzheimer's, Parkinson, even metabolic syndrome, autoimmune condition, psychiatric, which I gave an example earlier, fibromyalgia as well, and definitely cancer. Now this figure is again reiterating that mitochondrial dysfunction is the basis of constellation of metabolic abnormalities, ranging from insulin resistance to diabetes, abnormalities in fatty acid metabolism. Like even if you're looking simply at the pathway, you won't see that, but once you realize that a part of that pathway is happening in the mitochondria, it makes sense. I also like this figure. It's actually saying a similar thing, but look at the orange dots. These are the ones that we are now aware are directly linked to mitochondrial dysfunction. Alzheimer's, Parkinson, even autism. Okay. From exam perspective, please remember some of these disease conditions. Looking a little deeper, the example of the slide, which I actually added earlier in the session, that mutations in the mitochondrial DNA are now observed in certain disease conditions. Whether that's happening again, I try to keep an open mind, whether it's just environment or whether it's something else. I think you can make a fair guess. Okay. Important point, can we measure mitochondrial dysfunction? Would somebody like to comment? Has anybody heard or read about it? Not very important for this subject maybe, but you will learn about it when you go into the field of functional testing. That's another reason I shared that Krebs cycle diagram again with you, so that some of that stay with you even when you go in the clinic. So some of the tests that are listed on this slide are a measure of mitochondrial dysfunction. One is we can measure whether pyruvate or lactate levels are elevated in the blood. We can measure plasma or urinary coenzyme Q10 levels. We can measure red blood cell glutathione. That will give us an indication whether mitochondria are protected by free radicals or not. But it is the first one that is kind of popular among our community. And I think that window is open. I'll take a second to open up one of the reports. If you are interested, what does it look like? It was shared with me by one of my students. I would like not to divulge his details, so kindly ignore, just for the privacy's sake. Okay, so he shared with me just so that future learners can have a look. What happens in this OATS report, that it is popularly known as organic acid test. What we are able to measure here is those intermediary compounds of the mitochondrial pathway, that is ETC. Maybe at this stage, don't focus on the jargon of words that are coming your way, but just focus here. So we can look at lactate, citrate, isocitrate. So this is a urine test that we do. And when we measure that there is an excess of particular intermediary metabolite from ETC cycle coming in the urine, it gives me an indication that cycle is stuck at some place. Okay, that's why maybe somewhere in the long-term memory, remember succinate, citrate, and all those words. And then kind of this test help us understand that we need to support a person's mitochondria. The report is much more detailed. We even go into neurotransmitter metabolism, protein metabolism, even details of fatty acid metabolites. Okay. Don't feel overwhelmed then. If you have that pathway from nutritional biochemistry saved in your notes somewhere. So one of those things which I say that you need to save it in the notes somewhere. Was that helpful? Yes, Annie, you are right. This test actually very directly shows that a person's mitochondrial function is impacted. And I remember, you know, when I'm in the clinic, students want to test it. And a couple of my students last year, they did it on their own. And then they had a meeting with me as well as the lab representative so that they can discuss about it. In your to-do things later, have it somewhere. Okay. Coming up to again refresh our understanding of the nutrients that are essential for mitochondria. This diagram is colorful. First look at it and I don't know what's happening here. It is so messy. But then you realize, okay, this is the ETC. Look at the first set of complex, 1. The nutrients that are imperative here are vitamin B2, selenium, cofactor Q10. And I'm sure one of you is going to ask me, what is caffeine doing here? So one of the reasons we feel energetic when I had that coffee in between the lecture was that transiently it supports mitochondrial health. Then look at the second complex, very small here. Nutrients that are needed are Q10, vitamin E, selenium, B2. Further, complex 3 has few other nutrients, and so it's kind of a very good summary. If you can save it and in future you can layer it with your understanding of organic urine test and see exactly where the hindrance in a particular person and which nutrients need to be given immediately or through dietary intervention. Okay. So now we want to look at which are those major nutrients that support and that are known to prevent mitochondrial dysfunction. It is not an exhaustive list. The way I have included this couple of slides is based on whatever research-based data I could collate. So one I found was coenzyme Q10. Somebody coming to you with fatigue, energy, and you can't nail it down to a particular disease or a micronutrient deficiency such as iron, then look at coenzyme Q10. Immediate relief, people do feel energetic. Some people may have issues with fat metabolism, and there both lipoic acid and acetyl-carnitine, they support mitochondrial ATP production by increasing metabolisms of fat. Another reason for carnitine to be so popular for weight loss, even for muscle building, people who are trying to have extended physical activity levels. So that's one of the pathways. Resveratrol can also protect because it is, being an antioxidant, it is initiating that signaling pathway. Citrulline is that gene expression pathway, which is modulated by some of the antioxidants because it upregulates the function of antioxidants. Rest nutrients, I'm sure you're all aware of. You know the reasons also. So far, we have come to session 12. We all have learned so much and hopefully understood the amazingness of these micronutrients. Would anybody like to point out some other nutrients that you think may be important for mitochondria? Any other question, any other comment? Because that's all for today from my side. This diagram is taken from this reference and it should be there in the slide. Yeah, this one. So, Myhill and Booth and McLaren Howard. If you want to get a better picture because it makes so much sense. Okay, good. I noticed that students find this session really interesting, and as it kind of makes sense, the new knowledge that we are learning, the deeper we are going in our journey to understand ourselves, our relation with nature, environment, universe, this information kind of builds a lot. But what I would like to see more is this information being utilized once you reach clinic and start when you're looking at disease etiology, start looking at this aspect as one of the causes. And okay, thank you everyone. One question by Tracy. With the diseases mentioned that are relative to impaired mitochondrial function, can we support or inhibit the disease progression with the nutrients? Good point. Yes, we can. Okay. I have read and seen a few cases where we can make a difference through supplements if we can pinpoint. And that's why I said watch that TED Talk. We can even reverse. Oh, Tracy, so sorry to hear about a friend who was recently diagnosed with MS. Again, if you haven't watched that TED Talk, please watch it today. And in fact, the nutrients can reverse the changes. That's what I learned when I saw this TED Talk many, many years ago. That's what I said. It's an old talk. It's a very slow talk, but it's made so much sense. Okay. Bye for now, guys. Have a good week.